Welcome to the AP Biology Final, Chapter 49, 50, and 51 by Jesse and Marine. Chapter 49. Chapter 49 is the nervous system. The key concepts we will be reviewing for this chapter will be 49.1. Nervous systems consist of circuits of neurons and supporting cells. 49.2. The vertebrate brain is regionally specialized. 49.3. The cerebral cortex controls voluntary movement and cognitive functions. 49.4. Changes in synaptic connections underlie memory and learning. 49.5. Nervous systems disorders can be understood in molecular terms. Concept 49.1. Nervous systems consist of circuits of neurons and supporting cells. An overview of the nervous system. Nervous systems perform the three overlapping functions of sensory input, integration, and motor output. Nervous systems are networks of neurons with intricate connections. Nerves channel and organize information that flows along specific routes throughout the nervous system. The nature of nerve cells. Every cell has a voltage or membrane potential across its plasma membrane. Changes in the membrane potential of the neuron give rise to nerve impulses. Nerve impulses propagate themselves along an axon. Chemical or electrical communication between cells occurs at synapses. Neural integration occurs at the cellular level. The same neurotransmitter can produce different effects on different types of cells. So here we have a picture of your regular nerve, human nerve, and as you can see, what happens is the nerve gains a message to the dendrite, which travels through the axon and ends at the axon terminal where the message is delivered. Animals and nervous systems, clusters of neurons perform specialized functions. Such clustering is absent in the ceridians, the simplest animals with nervous systems. In most ceridians, a series of interconnected nerve cells formed a diffused nerve net, which controls the contraction and expansion of the gastrovesicular cavity. In more complex animals, the axon of multiple nerve cells are often bundled together, forming nerves. As the organism gets larger, the more complex the nervous system is. The spinal cord transports information to and from the brain and produces reflexes, automatic responses to stimuli. And here we have a picture of the diversity of nervous systems between ceridians, ectoderms, flatworm, annelid, arthropod, mollusk, and chordate. And as you can see, they're all completely different, but they all have a similar structure except for the ectoderm, because it has a nerve ring and a radical nerve, compared to a ceridian, which just has a nerve net throughout its whole body. Gray matter consists of neuron cell bodies, dendrites, and unmammalian axons. White matter is named for the white color of the mailing sheets of the bundle of axons. Astrocytes are supporting glia with various functions. Some facilitate information transfer at synapses and increase blood flow to activate neurons. Radical glia guide with embryonic growth of neurons. Sensory information is carried to the CNS along different neurons. Afferent neurons to the peripheral ectoderm carry instructions to muscles, glands, and endocrine cells. The motor system carries signals to skeletal cells. The autonomic nervous system maintains the internal environment by its control over smooth and cardiac muscles and various organs. The sympathetic division accelerates the heart and metabolic rate, generating energy. The parasympathetic division creates signals that enhance self-maintenance, activates that conserve energy. The enteric division includes networks of neurons that control the secretion of the digestive tract. Concept 49.2. The vertebrate brain is regionally specialized. Evolution and diversity of nervous systems. The ability of cells to respond to the environment has evolved over billions of years. Nervous systems show diverse patterns of organization. The vertebrate nervous system. Vertebrate nervous systems have central and peripheral components. The divisions of the peripheral nervous systems interact in maintaining homeostasis. Embryonic development of the vertebrate brain reflects its evolution from 3 anterior bulges of the neural tube. As vertebrate embryo develops, the forebrain, midbrain, and hindbrain become evident as 3 bilaterally symmetrical anterior bulges of the neural tube. And here we have a picture of the embryonic brain regions compared to the brain structures present in adults. And they all have the same, as you can see, the embryo with 1 month has the 3 main parts of the brain, or what used to be the 3 main parts of the brain, compared to the adult, which has 5 parts. The same thing with the 5-week-old. So from the forebrain, the cerebrum and the thalamus, hypothalamus, and epithalamus come from the forebrain. And the midbrain, it just stays the same. It's connected to the part of the brainstem. And the pons and medulla come from the midbrain. The hindbrain, excuse me. By the 5th week of embryonic development in humans, the 3 primary bulges have formed 5 brain regions, so that's what we just talked about. The hindbrain also gives rise to another major brain center, the cerebrum. The telophyon is the region of the forebrain that gives rise to the adult cerebrum. Rapid growth of the telophyon during 2nd and 3rd months causes the cerebral cortex to extend over and around the brain. The brainstem functions in is homeostasis, coordination of movement, and conduction of information. The adult brainstem is made of midbrain, pons, and medulla oblongata. The medulla and pons assist in the transfer of information between the PNS and the midbrain and forebrain. They also help coordinate large-scale body movements like running. The midbrain contains centers of receiving and integrating several types of sensory information. All sensory axons involved in hearing either terminate in the midbrain or pass through and enter their way through the cerebrum. In non-mammalian vertebrates, portions of the midbrain form prominent optic lobes that may be the only visual centers. In animals, vision is integrated in the cerebrum, not the midbrain. The mammalian midbrain coordinates visual reflexes such as the peripheral vision reflex, in which the head turns to the approaching object without forming an image of it. Signals from the brainstem affect attention, alertness, appetite, and motivation. Arousal is a state of awareness of the external world. Sleep is a state in which external stimuli are received but not consciously perceived. The reticular formation is a diffused network of neurons in the core of the brain. In the brainstem, acts as a sensory filter to choose which information reaches the cerebral cortex. The more info the cortex receives, the more alert and aware one is. The brain often ignores certain stimuli while actively processing other inputs. The pons and medulla have centers that cause sleep, and the midbrain has a center that causes arousal. All birds and mammals show characteristics of sleep-wake cycles regulated by melatonin. Melatonin is a hormone produced by the pineal gland. Melatonin is also synthesized from the amino acid serotonin, which may be the neurotransmitter of the sleep-producing centers. Brainwave frequencies change as the brain processes through distinct stages of sleep. The cerebellum coordinates movements and balance, and also is involved in learning and remembering motor skills. The embryonic dicynone evolved into three brain regions: the thalamus, hypothalamus, and epithalamus. The epithalamus includes the pineal gland and the source of melatonin, and also contains one of several clusters of capillaries that generate cerebrospinal fluid from blood. The thalamus is the main input center of sensory information going to the cerebrum. Incoming information from all the senses is sorted in the thalamus and sent to the appropriate cerebral centers for further processing. Now, the hypothalamus. It is one of the most important brain regions for the control of homeostasis. And homeostasis is pretty much when your body balances itself and regulates itself. So if you just worked out really hard and you're sweating a lot, homeostasis comes in and it balances your body temperature so that you can cool down. The hypothalamus contains the body's thermostat as well as centers for regulating hunger, thirst, and other basic survival mechanisms. In other organisms, circadian rhythms in mammals rely on a biological clock, or circadian, my bad. SCN are a group of neurons in the hypothalamus that coordinate mammal circadian rhythms. And circadian rhythms is pretty much how our own body, it's our body's internal clock that is based on the 24-hour system. That's why we have 24 hours in a day. And our bodies have adapted to that as the world has changed. In mammals, information processing is largely centered in the cerebrum. The cerebrum develops from the embryonic telencephalon on an outgrowth of the forebrain that arose early in vertebrate evolution as a region supporting olfactory reception, as well as auditory and visual processing. The cerebrum is divided into right and left cerebral hemispheres. The left cortex receives information from and controls the movement of the right side of the body and vice versa. The corpus callosum enable the left and right cerebral cortices to communicate. Concept 49.3. The cerebral cortex controls voluntary movement and cognitive functions. Each side of the cerebral cortex is divided into 4 lobes: frontal, temporal, occipital, and parietal, which all have specialized functions. Functional areas include primary sensory areas, which receive and process specific types of sensory information, and association areas, which integrate information from parts of the brain. Sensory information is directed to primary sensory areas located in different lobes of the cortex. Associate areas near the primary sensory areas integrate the different aspect of sensory inputs. Here you'll see a picture of the brain and how the 4 different lobes and what they control. The frontal lobe is responsible for speech, motor cortex, and the temporal lobe is responsible for smell and hearing. Parietal, taste, speech, reading, and some auditory association area. And for the optical, that's for vision, which is in the very back of the brain. Broca's area is in the frontal lobe is involved in speech generation. Wernicke's area is the posterior temporal lobe is involved in speech comprehension. Letterization results in 2 hemispheres, differing in function. The left hemisphere usually becomes specialized for language, math, and logic. The right hemisphere is involved in pattern recognition and nonverbal thinking. The amygdala, hippocampus, and olfactory bulb form a ring around the brainstem called the limbic system. The amygdala functions in creating emotional memories, and the prefrontal cortex in making is important in the experience of emotions, temperament, and decision making. And here you'll see a picture of Broca's area, which again is responsible for the speech generation, as we saw.
Concept 49.4 Changes in synaptic connections underlie memory and learning. Regulated gene expression and signal transduction establish the overall structure of the nervous system. In the first of two processes, neurons compete for growth factors, which are produced in limited quantities by target tissues. Cells that do not reach their targets undergo programmed cell death. Half the neurons formed in the embryo are eliminated. The second process is synapse elimination, in which numerous synapses are formed, more than required for its proper function. The activity of the neuron stabilizes synapses and destabilizes others. By the end of embryogenesis, neurons have lost, on average, half of their initial synapses. The capacity for the nervous system to be changed in response to activity is called neuroplasticity. We hold information for a certain period of time in the short-term memory, which is released if it becomes irrelevant. Knowledge that is retained longer is present in the long-term memory. Information can be recalled into the short-term memory from the long-term memory. For long-term memories, the links in the hippocampus are replaced with more permanent connections within the cerebral cortex. People who have suffered damage to the hippocampus are unable to retain new lasting memories. Motor skills are learned by repetition. Memorization requires changes in the strength of neural connections. Long-term potentiation is the lasting increase in the strength of synaptic transmission. LTP requires a presynaptic neuron that releases the excitatory neurotransmitter glutamine. LTP requires two types of glutamine receptors that artificially activates that particular receptor. Concept 49.5 Nervous system disorders can be explained in molecular terms. Many disorders, such as schizophrenia and drug addiction, cause more deaths combined than heart disease or cancer. Many disorders concerning mood or behavior can be treated with drugs or medicines. Nervous system disorders result from chemical or anatomical changes in the brain. Only a portion of the disorders are caused by genetic contributions. The other significant contribution comes from environmental factors. About 1% of the world's population is afflicted with schizophrenia, a severe mental disturbance that causes the patient to lose the ability to distinguish reality. Evidence suggests that schizophrenia affects neuronal pathways that use dopamine as a neurotransmitter. Bipolar disorder involves mood swings from high to low and consists of genetic and environmental components. Individuals affected by major depressive disorder lose interest in once enjoyable activities for them. It affects twice as many women as it does men. The manic phase of bipolar disorder is characterized by high self-esteem and productivity. The depressive phase comes with lowered ability to feel pressure. Concept 50.1 Sensory receptors transduce stimulus energy and transmit signals to the central nervous system. A sensory pathway begins with sensory reception, the detection of a stimulus by sensory cells. Sensory cells in organs, and the structures within sensory cells, are called sensory receptors. The conversion of a physical or chemical stimulus to a change in the memory potential of a sensory receptor is called sensory transduction, and the change in membrane potential is known as receptor potential. Many sensory receptors show extreme sensitivity and can detect the smallest unit of stimulus possible. Sensory information is transmitted through the nervous system in the form of nerve impulses or action potentials. The magnitude of a receptor potential controls the rate at which action potentials are produced by a sensory receptor. Many sensory neurons spontaneously generate action potentials at a low rate. Circuits of neurons process the action potential to generate the perception of stimuli. Perceptions include the five senses and do not exist outside of the brain. We distinguish stimuli by the connections that link sensory receptors to the brain. The brain distinguishes sensory stimuli based on where action potentials arrive in the brain. Amplification is the strengthening of stimulus energy during transduction, which often requires signal transduction pathways involving second messengers. Sensory adaptation, the undergoing of decrease in responsiveness, enables the detection of changes in environments that vary on stimulus intensity. Sensory receptors are divided into five categories: mechanoreceptors, electromagnetic receptors, thermoreceptors, and pain receptors. Mechanoreceptors respond to mechanical energies such as pressure, stretch, motion, sound, and touch. Chemoreceptors respond to chemical stimuli. General chemoreceptors transmit information about the total solute concentration of a solution, while specific chemoreceptors respond to specific types of molecules. Internal chemoreceptors respond to compounds such as carbon dioxide and amino acids. Electromagnetic receptors detect electromagnetic energy such as visible light, electricity, and magnetism. Photoreceptors respond to visible light and are often organized into eyes. Thermoreceptors respond to heat or cold and help regulate body temperature. Mammals have differentiated thermoreceptors, each for a particular temperature range. Pain receptors, or nociceptors, are a class of naked dendrites on the epidermis. Nociceptor density is the highest in skin, but pain receptors are associated with other organisms as well. Concept 50.2. The mechanoreceptors responsible for hearing and equilibrium detect moving fluid or settling particles. Statocysts are mechanoreceptors that function in an invertebrate sense of equilibrium. It consists of a layer of ciliated receptor cells surrounding a chamber that contains one or more statoliths, grains of sand or other dense granules. Many invertebrates have a general sensitivity to sound, although specialized structures for hearing are less common than gravity sensors. Many insects have localized ears, a tympanic membrane stretched over an internal air chamber. Sound waves stimulate the membrane and results in nerve impulses that are transmitted to the brain. In hearing, the ear converts the energy of sound waves to nerve impulses that are sent to the brain as sound. Moving air causes the tympanic membrane to vibrate. The three bones of the middle ear, the malleus, stapes, and incus, transmit the vibrations to the oval window, a membrane on the cochlea's surface. When the stapes vibrates against the oval window, it creates pressure waves in the fluid of the cochlea. The fluid causes the basilar membrane and attached hairs to vibrate up and down. Mechanoreceptors in the hair cells respond to the bending by opening or closing ion channels in the plasma membrane. Pressure waves travel through the vestibular membrane and pass around the apex of the cochlea. They dissipate as they strike the round windows and resets the apparatus for the next vibrations. The ear conveys information to the brain about two important sound variables: volume and pitch. The volume of sound means the amplitude of the sound wave. The pitch is a function of a sound wave frequency, the number of vibrations per unit time. The cochlea can distinguish pitch because the basilar membrane is not uniform along its length. Several organs in the mammalian ear detect balance, body movement, and position. The utricle and the saccules each contain a sheet of hair cells that project into a gelatinous material. Present in the cells are many small calcium carbonate particles called otoliths. When your head is tilted, the otoliths press on the hairs that protrude into the gel and signals your brain that the head is tilted at an angle. Otoliths also respond to acceleration. Fishes lack cochlea, eardrums, and external openings. Water vibrations caused by sound waves are conducted through the skeleton to a pair of inner ears, which sets otoliths in motion and stimulating hair cells. Most fishes and aquatic amphibians have a lateral line system along both sides of their body. In terrestrial vertebrates, the inner ear has evolved as a main organ of hearing and equilibrium. Birds also have a cochlea. Sound is conducted from the tympanic membrane by a single bone. Concept 50.3. The sense of taste and smell rely on similar sets of sensory receptors. Many animals use chemical senses to find mates, recognize territory, and to help navigate during migration. The perceptions of gustation, taste, and olfaction, smell, both rely on chemoreceptors. The receptor cells for taste in mammals are modified cells organized into taste buds. The receptors in taste buds are responsible for recognizing 5 types of tastings. 4 represent the familiar taste perceptions: sweet, sour, salty, and bitter. The fifth, called umami, is elicited by the amino acid glutamate. Any region of the tongue with taste buds can detect any of the 5 types of taste. The receptors fall into 2 categories, each evolutionary related to receptors for other senses. The sensation of sweet, umami, and bitter taste requires a G protein coupled receptor, or GPCR. Signal transduction to sensory neurons is similar for all GPCR type receptors. Unlike the other identified taste receptors, the receptor for sour tastants belongs to the TRP, transient receptor protein family. For decades, researchers were unsure whether a taste cell could have more than one type of receptor or whether each taste cell had a single receptor type, programming the cell to recognize only one of the 5 tastes. In 2005, Ken Mueller identified the family of bitter taste receptors. Using a cloned bitter receptor, he was able to genetically reprogram gustation in a mouse. Mueller found that an individual taste cell expresses a single receptor type and transmits action potentials to the brain representing only one of the 5 tastes. Olfactory receptor cells line the upper portion of the nasal cavity. In olfaction, unlike gustation, the sensory cells are neurons. In mammals, olfactory receptor cells line the upper portion of the nasal cavity and send impulses along their axons directly to the olfactory bulb of the brain. The receptive ends of the cells contain cilia that extend into the layer of mucus coating the nasal cavity. When an odorant diffuses into this region, it binds to a specific GPCR protein called an odorant receptor, OR, on the plasma membrane of the olfactory cilia. In olfactory cells, cyclic AMP opens channels in the plasma membrane that are permeable to both sodium and calcium carbonate ions. The flow of these ions into the receptor cells depolarizes the membrane, generating action potentials. Humans can distinguish thousands of different odors, each caused by a structurally distinct odorant. Cells with different odorant selectivities are interspersed in the nasal cavity, but their axons sort themselves out in the olfactory bulb of the brain. Concept 50.4. Similar mechanisms underlie vision throughout the animal kingdom. Most invertebrates have some kind of light detecting organ. The ocelli of planarians are amongst the simplest photoreceptors. Ocelli are surrounded on 3 sides by a layer of darkly pigmented cells that block light. Light shining on the planarian stimulates photoreceptors in each ocellus only through the opening where there are no pigmented cells. Compound eyes are found in insects and crustaceans, and in some polychaete worms. A compound eye consists of several thousand light detectors called ommatidia, each with its own light-focusing lens. Each ommatidium detects light from a tiny portion of the visual field. A compound eye is effective at detecting movement, an important adaptation for flying insects and small animals threatened with predation. Insects also have excellent color vision, and some can see into the ultraviolet range of the electromagnetic spectrum. Among invertebrates, single lens eyes are found in some jellies and polychaetes, as well as spiders and mollusks. The globe of the vertebrate eye, or eyeball, consists of the sclera, a tough white outer layer of connective tissue, and a thin pigmented inner layer called the choroid. At the front of the eye, the sclera becomes the transparent cornea, which lets light into the eye and acts as a fixed lens. Just inside the choroid, the retina forms the innermost layers of the eyeball and contains layers of neurons and photoreceptors. The human retina contains rods and cones, two types of photoreceptors that differ in shape and in function. Rods are more sensitive to light but do not distinguish colors. Cones provide color vision but contribute very little to night vision. Rods are more sensitive to light but do not distinguish. There are three types of cones. Each has a different sensitivity across the visible spectrum, providing optimal response to red, green, or blue light. The distribution of rods and cones varies across the human retina. The fovea, the center of the visual field, has no rods but an extremely high density of cones, about 150,000 cones per square millimeter. The ratio of rods increases with distance from the fovea, with the peripheral regions having exclusively rods. In humans, color blindness is due to alterations in the genes for one or more photopsin proteins. Visual processing begins with rods and cones synapsing with neurons called bipolar cells. In the dark, rods and cones are depolarized and they continue to release the neurotransmitter glutamate at these synapses. In the light, rods and cones hyperpolarize, shutting off the release of glutamate. Three other types of neurons contribute to information processing in the retina: ganglion cells, horizontal cells, and amacrine cells. Horizontal cells and amacrine cells help integrate the information before it is sent to the brain. Light must pass through several layers of neurons to reach the photoreceptors. All the rods or cones that feed information to one ganglion cell form a receptive field, the part of the visual field to which the ganglion can respond. The two optic nerves that transmit sensations from the brain to the eyes meet at the optic chiasm near the end of the base of the cerebral cortex. At the optic chiasm, sensations from the left visual field of both eyes are transmitted to the right side of the brain, and sensations from the right visual field are transmitted to the left side of the brain. Additional neurons carry the information to higher order visual processing and integrating centers elsewhere in the cortex. 30% of the cerebral cortex help formulate what we see.
Concept 15.5. The physical interaction of protein filaments is required for muscle function. Muscle cells function relies on microfilaments, which are actin-containing components of the cytoskeleton. Microfilament movement brings about contraction, whereas muscle extension occurs passively. A skeletal muscle contains of a bundle of long fibers running parallel to the length of the muscle. The microfilaments are composed of two kinds of myofilaments, thin and thick filaments. Thin filaments consist of two strands of actin and one strand of regulatory protein coiled around each other. Thick filaments are staggered arrays of myosinous molecules. According to the sliding filament model of muscle contraction, neither the thin nor the thick filament change in length when the sarcomere shortens. Instead, the filaments side past each other longitudinally, increasing the overlap between the thin and thick filaments. The sliding is based on the interaction between the actin and myosin molecules that make up the thick and thin filaments. Each myosin molecule has a long tail region and a globular head region. The tail adheres to the tails of the other myosin molecules that form the thick filament. The head of the center of the bioenergetic reaction that power muscle contraction, binding the hydrolyzing ATP and ADP, and inorganic phosphate. Hydrolysis of ATP converts myosin to a high-energy form that bind to actin, forming a cross binge and pulling the thin filament toward the center of the sarcomere. Each of the approximately 350 heads of thick filament forms and reforms about 5 cross bridges per second, driving filaments past each other. The energy required for continued contraction is stored in creatine phosphate and glycogen. The regulatory proteins, tyrosine and the trypsin complex, binds to thin filaments. In a muscle fiber at rest, tropomyosin covers the myosin binding sites along the thin filament and prevents the interaction of actin and myosin. The thin and thick filaments slide past each other and the muscle fiber contracts. Several diseases cause the paralysis of interfering with the excision of skeletal muscle fibers by motor neurons. A single skeletal muscle fiber contracts completely in a brief all or none twitch, or not at all. A whole muscle composed of mainly individual muscle fibers can contract to varying degrees. Greater contraction is due to variation in the number of muscle fibers that contract and variation in the rate at which muscle fibers are stimulated. A motor unit consists of a single motor neuron and all of the muscle fibers it controls. When a motor neuron produces an action potential, all the muscle fibers in its motor unit contract as a group. As more and more of the motor neurons controlling the muscle are activated, a process of recruitment increases the force developed by the muscle. Prolonged contraction of muscles can result in fatigue, caused by depletion of ATP and dissipation of ion gradients. Because muscle fibers are connected to bones by tendons and connective tissue, a contracting muscle fiber stretches these elastic structures, transmitting tension to the bones. Fiber types are classified either by the source of ATP used to power muscle activity or by the speed of muscle contraction. These fibers may have mitochondria, a rich body supply, and the large amount of an oxygen-storing protein called myoglobin that binds oxygen more tightly than hemoglobin does. Most human skeletal muscles contain both fast and slow twitch fibers, although the muscles of the eye and hand are exclusively fast twitch. In a muscle with a mixture of fast and slow twitch, the relative proportions of each genetically determined. Although cardiac muscle is straight like skeletal muscle, the structural differences between the skeletal and cardiac muscle fibers result in differences in their electrical and membrane properties. Cardiac muscle cells can generate their own action potentials without nervous system input. Smooth muscle lines on the walls of blood vessels and digestive system organs. Smooth muscle lacks the striations seen in skeletal and cardiac muscle. In smooth muscle, thick filaments are scattered through the cytoplasm, and thin filaments are attracted to structures cause dense bodies tethered to the plasma membrane. Smooth muscle contractions and relaxation are slower than the strained muscle. Smooth muscle lacks trophin complexes and tubules and has poorly developed SR. Concept 50.6. Skeletal systems transform muscle contraction into locomotion. Muscles work in concert with the skeleton to move an animal's body. The skeleton provides the rigid structure to which muscles can attach. So your skeleton is it is your backbone and the not exoskeleton, but it's your skeleton. It's the frame of your body. And the nervous system coordinates the functions of the two muscles in an autonomic pair. Skeletons function in support and protection as well as facilitating movement. In many animals, a hard skeleton protects soft tissues. Hardened support can be external in the form of exoskeletons, internal in the form of endoskeletons, and non-existent such as in fluid-based or hydrostatic skeletons. A hydrostatic skeleton consists of fluid held under pressure in a closed body compartment. Form and movement are controlled by changing the shape of this compartment. Along the ceridians, a hydra can elongate by closing its mouth and using contractile cells in the body, while it consists the central gastrovesicular activity. In flatworms, intestinal fluid is kept under pressure. It is kept as a skeleton and served as the main hydrostatic skeleton. In annelids, colloidal fluid acts as a hydrostatic skeleton. Earthworms use their hydrostatic skeleton to move by paralysis. Hydrostatic skeletons are adventurous in an aquatic environment and support crawling and burrowing in terrestrial animals. An exoskeleton is a hard encasement deposit of the surface of an animal. Many mollusks are enclosed in a calcium carbonate shell secreted by mantle. The joined exoskeleton of arthropods is composed of a cuticle with muscles attached to the interior surface. About 30 to 50% of the cuticle consists of keratin. Fibrils of keratin are embedded in a protein matrix, forming a composite material that combines strength and flexibility. Some crustaceans, such as lobsters, harden portions of their exoskeleton by adding calcium salts. With each growth spurt, an arthropod must shed its exoskeleton and provide a larger one. An endoskeleton consists of hard supporting elements held within the soft tissues of the animal. Sponges are reinforced by hard needles and inorganic material of soft protein fibers. Coordinate skeletons are composed of cartilage, bone, and some contribution of the two. Different modes of locomotion vary in energy cost. In all its modes, locomotion requires that an animal expend energy to overcome two forces that tend to keep it stationary: friction and gravity. Because water is buoyant, gravity poses less of a problem for swimming than for other modes of locomotion. Many insects of four-legged vertebrates use their legs as oars to push against the water. Squids and scallops are jet-propelled. Sharks and bony fishes move their bodies and tails from side to side, while whales undulate their bodies and tails up and down. For locomotion on land, powerful muscles and skeletal support are more important than a streamlined shape. When a quadruped walks, it keeps 3 feet on the ground to maintain balance. When an animal runs, all 4 feet may be off the ground briefly, but at running speeds, it is momentum more than foot contact that keeps the body upright. Crawling requires a considerable amount of energy to overcome friction, but maintaining friction balance is not a problem. Gravity poses a major problem for flight because wings must develop enough lift to overcome gravity's downward force. Flying animals are light with body masses ranging from less than a gram for some insects to 20 grams for the large flying birds. An animal's use of energy to move determines how much energy in food is available for their activities, such as growth and reproduction. The structural and behavioral adaptations that maximize the efficiency of locomotion increase an animal's evolutionary fitness. Chapter 51. Chapter 51 is animal behavior. The key concepts are 51.1. Discrete sensory inputs can stimulate both simple and complex behaviors. 51.2. Learning establishes specific links between experience and behavior. 51.3. Both genetic makeup and environment contribute to the development of behaviors. 51.4. Selection for individual survival and reproduction success can explain most behavior. 51.5. Inclusive fitness can amount for the evolution of a strict social behavior. Concept 51. A discrete sensory input is the stimulus for a wide range of animal behaviors. An animal's behavior is the sum of its responses to external and internal stimuli. In the mid-20th century, behavioral biologists developed the discipline of ethology, a scientific study of how animals behave in their natural environments. A fixed action pattern is a sequence of unlearned behavioral acts that is essentially unchangeable and once initiated is usually carried to completion. The FAP is triggered by an external sensory stimulus called a sign stimulus. A kinese is a simple change in activity or turning rate in response to a stimulus. A taxi is an oriented movement toward or away from a stimulus. Many fish, birds, and other animals undergo migration, a regular long-distance change in location. One hypothesis is based on the discovery of the manic jet, a magnetic iron ore in the heads of migrating fish and birds. One hypothesis suggests that animals are guided by magnetic field effects on photoreceptors in the visual system. The output of a circadian clock is a circadian rhythm, a daily cycle of rest and activity. Behavioral rhythms, such as migration and reproduction, are circadian rhythms linked to the yearly cycle of seasons. Not all biological rhythms are linked to the light and dark cycles in the environment, but animals may generate environmental stimuli that guide other animals' behaviors. So a stimulus transmitted from one animal to another is called a signal. The transmission and reception of signals constitute animal communication. Visual communication is the recognition of the sex and by appearance. Chemical communication is the transmission and reception of signals in the form of specific chemicals released by one and detected by the other. The opposite sex olfactory system, which again is smell. Tactile communication, or it's one sex to the opposite sex's presence. Auditory communication, the sex whether the opposite sex is the same species. The type of signal used to transmit information is closely related to an animal's lifestyle and environment. Most terrestrial animals are nocturnal, which make visual displays relatively ineffective and they rely on olfactory and auditory signals. Like birds, humans are diurnal and use mainly visual and auditory communication. Pheromones are especially common among mammals and insects and often relate to reproductive behavior. Pheromones may cause function in non-reproductive behavior. For example, when a catfish is injured, an alarm substance in the fish's skin disperses in the water, inducing a fright response in other fish. Pheromones can be ineffective to low concentrations. 1 centimeter squared of skin from a fatted minnow contains sufficient alarm substance to induce a reaction in 58,000 liters of water. Concept 51.2. Learning establishes specific links between experience and behavior. For behaviors such as fixed action patterns, taxes and pheromone signals in individual in the population almost the same behavior, despite the internal differences and environmental differences during development. Behavior is developmentally fixed in this way is called innate behavior. Environmental conditions can influence behavior through learning, the modification of behavior based on specific experiences. Habitation, one of the simplest forms of learning, is the loss of responsiveness to stimuli that convey little or no new information. Habituation prevents the wasting of time and energy on stimuli that are irrelevant to the animal's survival and reproduction, thus increasing an individual's evolutionary fitness. Imprinting the formation early in life that of a long-lasting behavioral response to a specific individual or object includes both learning and innate components. Imprinting is limited to a sensitive period, a specific developmental phase when certain behaviors can be learned. The tendency to respond to the outside world provides the imprinting stimulus to which the response will be directed. An organism's fitness may be enhanced by its capacity for spatial learning, establishing a memory that reflects the environment's spatial structure. Landmarks are location indicators occasionally used by animals. Learning leads to an animal modifying its behavior based on information from the environment. The ability to associate one environmental feature with another is called associative learning. In operant conditioning, an animal learns the associated one of its own behaviors with a reward or punishment and then tends to repeat or avoid that behavior. In classical conditioning, an afterbury stimulus becomes associated with a particular outcome. Although it was thought that animals could learn to link and stimulus to any behavior, this idea was proven incorrect. The most complex forms of learning involve cognition. The ability of one animal's nervous system to receive, store, process, and use information gathered by sensory receptors. Many animals besides humans and other primates display cognition. Insects can categorize objects according to abstract concepts. Problem-solving behavior is generally developed in primates and dolphins. Many animals learn to solve problems by observing the behaviors of other individuals. Development of some behaviors occurs in distinct stages.
Concept 51.3 Genetic makeup and environment both contribute to the development of behavior. Animal behavior is based in complex interactions between genetic and environmental factors. This finding contrasts with the popular conception that behavior is due to one or the other. The extent to which the offspring's behavior changes indicates how the social and physical environment influences behavior. For humans, the influence of genetics and environment on behavior can be explored by a twin study in which researchers compare the behavior of identical twins raised apart with those raised in the same household. Concept 51.4 Selection for individual survival and reproductive success can explain most behaviors. Optimal foraging theory views foraging behavior as a compromise between the benefits of nutrition and the costs of obtaining food, such as the energy expenditure and risk of predation while foraging. Natural selection should favor foraging behavior that minimizes the cost of foraging and maximizes the benefits. How mating behavior enhances reproductive success varies, depending on the species' mating system. In many species, mating is promiscuous with no strong pair bonds or lasting relationships. In species in which the mates remain together for a longer period, the relationship may be monogamous, one male mating with one female, or polygamous, one individual mating with many partners. Polygamous relationships may involve a single male and many females, polygyny, or a single female and many males, polyandry. Among monogamous species, males and females are often morphologically similar and difficult to distinguish based on external characteristics. Polygynous species are generally dimorphic, with males being larger and more showy. In polyandrous species, females are more ornamented and larger than males. The needs of young are an important factor constraining the evolution of mating systems. Most newly hatched birds cannot care for themselves and require a large, continuous food supply that a single parent cannot provide. In mammals, the lactating female is often the only food source for the young. The needs of young are an important factor constraining the evolution of mating systems. Most newly hatched birds cannot care for themselves and require a large, continuous food supply that a single parent cannot provide. In mammals, the lactating female is often the only food source for the young. Males provide no parental care in most mammal species. Certainty of paternity can influence mating systems and parental care. Certainty of paternity is much higher in species that engage in external fertilization when egg-laying and mating occur together. This finding may explain why parental care in aquatic invertebrates, fishes, and amphibians, when it occurs, is as likely to be by males as females. Sexual selection can take the form of intersexual selection, in which members of one sex choose mates on the basis of particular characteristics of the other sex, such as courtship songs, or intersexual selection, which involves competition among members of one sex for mates. A female that chooses a healthy male increases the chance that her offspring will be healthy. Males compete with each other by agonistic behaviors that determine which competitor gain access to resources. Game theory evaluates alternative strategies in situations where the outcome depends on each individual's strategies and also on the strategies of other individuals. Concept 51.5. Inclusive fitness can account for the evolution of altruistic social behavior. Individual survival and reproductive success are favored by selection, regardless of their effect on other individuals. Altruism is behavior that decreases individual fitness but increases the fitness of other individuals in the population. In honeybees, workers are sterile but labor on behalf of a single fertile queen. Individuals sometimes help others who are not their offspring. Inclusive fitness is defined as the total effect an individual has on proliferating its own genes by reproducing and by helping close relatives produce offspring. Hamilton's rule states that natural selection favors altruism when the benefit to the recipient multiplied by the coefficient of relatedness exceeds the cost to the altruist. Natural selection that favors altruistic behavior by enhancing the reproductive success of relatives is called kin selection. Some animals behave altruistically toward others who are not close relatives. This exchange of aid is called reciprocal altruism and is commonly used to explain altruism between unrelated humans. Game theory provides a possible answer in the form of behavioral strategy called tit for tat, in which one individual treats another in the same way it was treated the last time they met. Social learning is learning through observing others. Social learning forms the roots of culture, which can be defined as a system of information transfers through social learning or teaching that influences the behavior of individuals in a population. Cultural transfers of information has the potential to alter behavioral phenotypes and influence the fitness of individuals. Mate choice copying is a behavior in which individuals in a population copy the mate choices of others. Human culture is related to evolutionary theory in the discipline of sociobiology, whose main premise is that certain behavioral characteristics exist because they are expressions of genes that have been perpetuated by natural selection. The spectrum of possible human social behaviors may be influenced by our genetic makeup, but that is very different from saying that genes are rigid determinants of behavior. Because of our capacity for learning, human behavior is probably more plastic than the behavior of any other mammal. In human behavior, as in other animals, gene and environmental factors build on each other.